diving into the biomarkers, I would like to define some terms that I'll be using, starting from dementia. So what is dementia? Dementia is not a disease itself, but it's an umbrella term that refers to progressive decline in memory and other thinking abilities that impair some daily living. And under this umbrella, we found different types of dementia, including frontotemporal dementia, Lewy body dementia, vascular dementia, and the most common one that is Alzheimer's disease. So all these types of dementia are caused by different brain pathologies, so the underlying cause of this. And talking about Alzheimer's disease, so that it is defined neuropathologically with aggregated forms of amyloid, beta, and tau in the brain. And it's what the combination of both that defines Alzheimer's disease pathology. So, but Alzheimer's disease pathology, it's not the only cause of the symptoms, so the clinical symptoms, so all the memory problems and other thinking abilities problems. There are others also. So this is to make the point that there are different brain diseases, different brain pathologies that can cause very similar symptoms. So, and this is why we need biomarkers, so to be able to detect these underlying causes and actually being able to distinguish all these forms of pathology in the brain before death. So when people are still alive and we can distinguish them. But what is a biomarker? So a biomarker is a biological marker, and it's a test that informs about a biological state. For example, it can tell us about the healthy function of the body, so normal physiology, tells us about pathological processes, so diseases, and response to treatments, for example. So these are some common examples that you might be familiar with, are all biomarkers, such as blood sugar, cholesterol, blood pressure. And excitingly now, we also have biomarkers of the brain, so that can tell us something about the brain. And so it's very important, as I was mentioning, to distinguish all these brain pathologies that cause dementia and all these symptoms. So, but how do we do that? So, biomarkers for brain aging and Alzheimer's disease, for example, there's neuroimaging techniques like MRI and PET that give us a picture, an image of the brain. They're tested done through a lumbar puncture, so then we can test the cerebrospinal fluid. And also now, excitingly, we have also blood biomarkers. So, what we do with these biomarkers? So there are different types, and some of them are specific. They are testing for specific brain pathologies. So, for example, here you can see two images of the brain. One is for amyloid and one is for tau. Both measure with PET imaging, and they're both markers of Alzheimer's disease pathology in the brain. And these proteins can also be detected in the cerebrospinal fluid and blood. We also have non-disease specific biomarkers, for example, MRI, that don't tell us something about exact pathology underlying the symptoms, but can tell us very valuable information about, for example, in the case of MRI, what brain regions are affected. So, and to further highlight the importance of these biomarkers, I wanted to tell you about this large multi-site study that's called the IDEA studies, where over 18,000 patients undertook an amyloid PET scan. So all patients, when amyloid PET to detect this amyloid, it's Alzheimer's disease pathology in the brain, and we could see that in patients with dementia or a milder form of difficulties in memory and other thinking abilities had an amyloid PET scan that was positive, meaning there was some signs of Alzheimer's disease pathology in their brain. But a substantial percentage of these patients did not have an amyloid PET scan, suggesting that they were not on the Alzheimer's disease pathway, so that their symptoms were caused by other brain pathologies. So that, again, suggesting how important it is to have biomarkers to understand what is the cause of the disease, because a patient could present with very similar symptoms, so very similar memory problems or language problems, visual perception, and etc., but also have different underlying causes. So I talked about the importance of biomarkers to improve the diagnosis and to detect different pathologies in the brain, but they're also used for biological staging, for example. So here I'm going to show you on the top row some images of, so on the top row you'll see amyloid plaques, and on the bottom row tau, both measured using PET. And I'm going to start you showing an image of a cognitively healthy older adult, and where we don't see signs of the pathology in the brain. So as the disease progresses, then both amyloid and plaques and tau start accumulated in the brain and spread, and you can see here in the image of these the red colors regions is where the pathology is accumulating. And again, as the disease progresses, also the pathology spread in the brain. We can use biomarkers to monitor disease progression. And so here in this graph, you see on the horizontal line time, and on the left graph, amyloid and tau on the right, both again measured using PET. And each line is single participants, and then we can also see in the black line the mean. And we can see that both of them increase over time. So the disease over time, there's more and more pathology in the brain, and we can measure it now in vivo using biomarkers, specifically PET in this case. So we use all this also in research studies to understand disease mechanisms, and also for therapies, for eligibility, to test eligibility, and also in response to treatments. And here you can see PET images before and after treatment with anti-amyloid therapies. So you can see that these red colors are decreasing after treatment. And also we use biomarkers to monitor safety of these therapies. So Alzheimer's disease pathology can now also be measured using blood biomarkers. And so here in the left graph, you can see that in patients with a positive tau PET scan, that means that they had signs of tau in the brain. So that's one of the Alzheimer's disease pathology. And we can see that those that had a positive tau PET scan also had higher p-tau 217 in their blood, suggesting that they were concordant. And also on the right graph, we can see that when we could actually have an autopsy, and so we could see post-mortem what was exactly the pathology underlying the symptoms, those that had more advanced stages of disease of Alzheimer's disease pathology in the brain also had higher p-tau 217 there in their blood. And this is very important to validate also different biomarkers against each other, so we can see the concordance between blood and PET, but also with the gold standard, that is the autopsy, so when we can actually see the underlying pathology post-mortem. So far, I focus mostly on Alzheimer's disease pathology, but as I mentioned at the beginning, Alzheimer's disease pathology is not the only cause of dementia and cognitive impairment. So here I'd like to talk about Lewy body disease, so both Parkinson's disease and dementia Lewy bodies, that are Lewy body disease, they are characterized by the neural inclusions of Lewy bodies that are aggregates of another protein that's called alpha-synuclein. And excitingly now we can we can measure and test is alpha-synuclei in the cerebrospinal fluid through a lumbar puncture. So I want to show you some data on the study that was done right here at UCSF in autopsy-proven cases. And so people that we could actually see what was the pathology so the people that they had Lewy body disease, Lewy body pathology in their brain, and we could see that all patients with signs of Lewy body pathology in their brain had a positive biomarker, and also there was a high percentage also in cases with a more limited spread, and also almost all patients that did not have the pathology had a negative test. So again, going forward, talking about non-Alzheimer's biomarkers, I mentioned that there are also non-disease specific biomarkers that don't tell us any about the exact disease underlying it, but can tell us very valuable information. For example, MRI, so through an MRI scan. So here you can see on the far left an MRI scan of a person with a cognitively healthy, then patients with dementia Lewy bodies, Alzheimer's disease, and on the far right, frontotemporal dementia. And here, even though we don't have information through an MRI about the exact pathology, we can still appreciate the difference in the pattern of different brain volumes and different brain regions that are affected by these different diseases. And also in this other image, of these white regions that we can see, we can that are called white matter hyperintensities, and we can see vascular change in the brain. So to summarize, biomarkers have truly transformed Alzheimer's disease research, have improving our understanding of the disease, helping the diagnosis, and also monitor treatments. And also there's ongoing research, so thanks to all participants for that, and researching more on the blood biomarkers, importantly measuring also non-Alzheimer's disease pathologies and extending the research to the diverse populations. So now I'd like to take a moment to thank all the incredible people that are involved in research, researchers, clinicians, staff, and the research participants, because that's how we can research this. All this wouldn't be possible without the invaluable contribution of all research participants. So thank you very much.